Our guest speaker today is Dr. Sara Campinoti from the Roger Williams Institute of Hepatology Foundation for Liver Research. Sara is a postdoctoral scientist who uses tissue engineering to produce a model of human liver. Sara will take you on an in-depth journey into her research, why it is important, and the potential benefits it brings to transplant patients. Sara, welcome to Catalyst Magazine Live.
Thank you very much to Catalyst for a very kind invitation. I'm very excited to be here and to introduce myself and the topic I am interested about. So I'll tell you everything about tissue engineering. So first of all, who am I? As kindly introduced, I'm Sara Campinoti. I've done my PhD at the Francis Crick Institute and at UCL. This is the Francis Crick Institute, which you might have seen if you have ever been to St Pancras station in London. It's just the opposite entrance of the station. And if you haven't been, I usually recommend you to go and visit next time you're in London. It's a huge, beautiful research institution with a museum in the ground floor, which you can access free of charge, always with very interesting exhibitions about science. Now, as mentioned, I'm a postdoc at the Liver Tissue Engineering and Regenerative Medicine Laboratory at the Roger Williams Institute of Hepatology, and this is the building I'm working in. Before getting on and talk about tissue engineering and what it is, I wanted to introduce what PhD and postdoc really means, which kind of job they are. You may be familiar with this terminology, but you may not. So I just thought about to introduce to you what is actually a PhD. PhD stands for Doctor of Philosophy, or sometimes is referred as doctorate, and is the highest level of degree that a student can achieve in academic career. A typical PhD involves quite a series of tasks that the students have to carry out, such as literature review, to know what is already known in the field of study, conducting original research and collecting results, for example, of experiments, then producing a thesis that represents all these experiments, writing up, of course, the thesis and submit it, and then defend the thesis via oral examination, which is called viva. So you may wonder exactly what a PhD means in terms of for a person. Many people think that PhD are only for geniuses, while actually, I tell you, not at all. For sure, you have to be very passionate about your PhD research question and to be driven to explore it very deeply. But it's not only for genius. Anyone with an interest can really achieve and get a PhD in the future. Other people think that we need to discover something incredible during a PhD, like Nobel Prize sort of discoveries. That's also not true. Not all PhD researchers lead to an extraordinary discovery. Of course, some of them do, but if they don't, it's okay. You need to prove that you have performed an original piece of research and you understand how that fits in the field of research, but it doesn't have to be breaking the rule of science. And many people think that after a PhD, a person must work in academia, must become a professor. And this is actually also not true. Actually, PhD degrees are extremely valuable outside academia. So if you're interested in working in industry, companies, but also government or startup, a PhD is, again, a very good choice and will open up so many doors. So in conclusion, if you're interested in a topic, I would really recommend you to consider a PhD after your university studies. So I'll tell you a bit more about my journey. This is where I was born, my hometown, Arezzo, which is in Tuscany, in the center of Italy, and that's actually where I am at the moment. And I did my bachelor university in Bologna, so a bit north from my hometown, and this is actually a picture taken the very first day I went in a laboratory, which was one of the best days of my life, really. And then I moved to Milan to do a master's degree in pharmaceutical biotechnology, and this is the day in which I got graduated. And then I moved to London, where I did my PhD, as I told you, at UCL and the Francis Crick. My PhD focused on stem cell, regenerative medicine, and tissue engineering, so topics I'm still studying, and I'm going to talk to you about today. And finally, this is me, just this past Christmas, where I work as a postdoctoral scientist. And these are a few of my colleagues and my supervisors. So I'm very fortunate to work with a very nice bunch of people. So after this introduction, I want to tell you why I decided to study liver now as my postdoc. And to introduce to the topic of the liver, you might know it's an organ in our body, but it's a very fascinating organ. And I tell you this by starting to tell you a myth, a Greek myth about Prometheus. Prometheus, that is shown here in this ancient picture, was living in the Olympus together with Zeus and other Greek goddess, and decided to gift the humans the fire, basically stole fire from the Olympus and brought it to human. But Zeus was very against it. He didn't want to give the fire to human. So when he found out that Prometheus betrayed him, he decided to punish him. And the punish was to chain Prometheus in the slope of a very big mountain. And every day, a very hungry eagle was going to Prometheus and eat his liver. So the Greek already knew that the liver could regrow, of course not overnight, but they thought that was a possibility. So basically every day this eagle was going to attack Prometheus and eat his liver, because the liver can regrow. The day after would have been the same and the same and the same forever. So this was a very terrible punishment for Prometheus, but it's very interesting to see how the liver capacity was already known. So the liver is master of regeneration, so can regrow in the organ, as an organ in our body, and it does very, very important function. One of the most important functions is to cleanse our blood from all waste and, for example, drug after we've taken them. It also produces very important enzymes, and they are released in the intestine, and they're called bile. And it stores energy in form of a very complex sugar that is called glycogen. So that's the function of the liver when it's healthy, and we know the liver can partially regrow. But there are series of injuries that can cause liver disease, and when this happens, the liver cannot repair itself, and we can have very big complications. So first of all, liver disease can be caused by, for example, excessive alcohol consumption, bad diet or fatty diet, abuse of drugs, and infections such as viral infection that we call hepatitis. All these insults eventually cause the liver that cannot cope anymore with all the stress, and liver disease, of course. Liver disease untreated eventually can progress to cirrhosis, that is basically not functioning anymore of the liver, or liver cancer. And why would I want to study liver, and why do I think it's very important to study liver right now? I'm going to show you with this graph. So as you can see here in the graph, there is a percentage of disease mortality throughout the year from the 1970 to the 2010. Many of the diseases that we know that are very dangerous, for example, circulatory or ischemic heart disease, so diseases that occur in the heart, or diabetes and even tumor, they are decreasing. So the mortality rate of all these diseases are decreasing. But as you can see here with this red line, disease mortality caused by liver disease are hugely increasing, and an increase of up for 400,000% since 1970. So this is very huge increase, and is a very important health problem in the UK and around the world. That's why I think we should really study liver disease. How can we cure a diseased liver? Well, unfortunately, today there are no drug or treatments that can stop liver disease, and that's probably why the curve is so high. But the only restorative treatment for end-stage liver disease is liver transplantation. You're probably familiar about the concept of transplantation. So a donor's healthy liver can be transplanted to a recipient person that is affected by liver disease, so the liver is removed and a new liver can be put in. However, liver transplantation is not that easy to be done. In fact, as you can see in this graph, in the light blue and in the dark blue, they are respectively the organ donors and the transplantation performed. So we have, this is in America, but it's a graph that can be applied anywhere in the world. We have quite a good ratio of transplanted organs. However, there are way more people awaiting for organs that we can cope. So in red, there is the line of patients awaiting for a liver transplant. So we don't have enough liver to be transplanted for the amount of people that they actually need. So this is, we call it shortage of organ, shortage of any organ for transplantation, but in this case, I'm talking about the liver. So scientists are really trying to find a way to remove this shortage by building organs in the laboratory. So what's the general principle about this is that we get from a patient, for example, cells that we can grow them in culture. We can make them high numbers. So from a few cells that we take from a patient, we can actually make billions. Then if we provide the cells a very good stimuli, for example, molecule that recapitulates the 3D environment of the organ, protein and other food and molecules that can stimulate the cells, sometimes even mechanical stimuli. So if we think about muscle, you will need a little bit of electricity to be functional. We can then make very complex 3D cultures that we can assemble somehow, and this is what I'm going to talk about in the laboratory, to then be transplanted back to the patient. So this is reality is happening right now. Many laboratories are studying how to do this, to provide a new organ to give to patient, and is what tissue engineering is studying. So the definition of tissue engineering is that it's a discipline that uses a combination of cell engineering, materials, and factors to restore, maintain, improve, or even replace different biological tissue, such as the liver, for example. But how can we create organ in a laboratory? So we need three major components: a scaffold, cells, and molecules. So today I'm going to talk about these three different components and how by combining them three, we could actually get tissue engineered organ to give it to patients. So first of all, let's talk about the cells. We need the right cells to be able to do this. Every organ of our body has different types of cells that are responsible of the organ function. If you think about it, cells in the brain are very different from cells in the liver or in the muscle, right? However, in most of the cases, it's very difficult to obtain the cells from an organ in the laboratory. First of all, because we don't have access to certain organs, and second, because it's very difficult to grow them. So a possible solution is offered by stem cell. Stem cells are undifferentiated cells, which have two major characteristics that you might remember from class. They can produce any kind of cells in the body in a process that we call differentiation, and they can also self-renew. So these are the two major characteristics. What are these? Why these characteristics are important? Well, with self-renew, we maintain the stem cell pool. So we will always have a pool of stem cells ready to be differentiated, because with the differentiation, we can replace dead or damaged cells through life. So we have several stem cells in our organ that do this job, but we can also have them in the laboratory. So to push them to do this in the laboratory. So which type of different stem cell do we have available? Well, we have embryonic stem cell, induced pluripotent stem cell, or adult stem cell. Let's have a look at the three of them and how we could use them in tissue engineering. So embryonic stem cell, or ES, are taken from the inner cell mass or a blastocyst, which is a very early stage in the embryonic life. These cells, when grow them in the laboratory in a medium that is this pinkish fluid with a lot of nutrients and molecules, can be then pushed to differentiate into basically any cell type of the body, because the cells come from the embryo that's not been developed yet. So we say that embryonic stem cells are pluripotent, so they can really dice to many different types of cells. Then we have induced pluripotent stem cells, in which we derive them by taking cells from the body, for example, from the skin. We can reprogram them to become stem cells. So we call them induced for this reason, because we induce them in the lab to revert, to become stem cell from non-stem cell. We can grow them in the laboratory, and again, we can differentiate them in so many types of cells. And even in this case, we call them pluripotent, so they can give rise to many different cells. Finally, we have adult or tissue stem cells. These are found in a series of organs in our body, from skin to eyes to even brain, intestine, muscle, lungs, so many organs, of course, liver, and in the bone marrow we find the blood stem cell. So these are very advantageous because they are a bit more, have less restrictions in terms of ethical compared, for example, an embryonic stem cell. However, they have a limitation. Because they come already from an organ, they can only give rise to the tissue, the cells of that tissue. So for example, intestinal stem cell will never produce neurons, will only give rise to intestinal epithelium. Neural progenitor, for example, to neuron, and stem cell in the bone marrow for the blood can give rise to the different blood cell type. For this reason, we call them multipotent, so they can differentiate in several cell types, but not all. The advantage of all these stem cells is that we can grow them in numbers in the lab, in culturing cells we also call in tissue culture conditions, and they can be transformed. So we can have a very good high number of these. As you can see, we can fill up incubators, which is where the cells grow. This is a picture of me in the lab growing some stem cell from the liver. So definitely with stem cell, we have the right cells to create organs in the laboratory for the capacity of obtaining very high number. What about the scaffold? Do we have a solution to give rise to like a structure for having this organ to be transplanted into patient? So to build an organ, we need the 3D structure, the backbone. And we like to call it scaffold to imagine sort of like a building that is being built. So the scaffolding, when we build, like, for example, a skyscraper, is very important because it gives you already the structure, more or less, of what will be the building next. And same for an organ, same for tissue engineering. We have different possibility here, and the future will only tell us what will be the best strategy. But I want to introduce a couple of strategies that the scientists are exploring. 3D printing is definitely one of the most promising. 3D printing technology is already very available in our daily life and is used for many applications, like for building these beautiful castles and models, toys, and even, technical appliances that we have in the house. However, 3D printed whole organs for transplantation, unfortunately, are not yet an option, although there is many research that is happening in many different organs. First of all, of course, liver and heart, but also lungs, gut, and even ears, for example, for giving it to people that unfortunately may have lost 1 year because of, for example, war, if you think about it, or bombing. So it's very, very interesting approach that hopefully in the future will be available to many. One of the main issues that is stopping 3D printing to get into clinic is to find a bioink and materials that are compatible with the cell survival and the culture of these organs, for example, using bioreactor. But research is happening. And in the meantime, scientists are working on a smaller scale, for example, for modeling a disease or testing some drug or testing some biology hypothesis. And this smaller scale of 3D printed organs are called organ on a chip. You may have heard them. Organ on a chip are printed mini version of organs that use a microfluidic device. This is how they look like. They're super tiny, so only a few centimeters big. But inside there are cells, most of the time stem cells, that can recapitulate the organ function and can allow us to test, for example, drug toxicity or how the organ works. And very interesting, these days, there are many more studies about multi-organ on a chip. So imagine these being slightly bigger and more interconnected, where you can have a portion of a chip with some lung cell and a portion of heart cell, bone cell, kidney, liver, and gut. For example, you could give an aerosol drug, maybe anti-COVID, for example, and they could test the effect of this drug into many different cell types of different organs. So this is extremely fascinating. We could measure how a drug affects the heart conduction or gets absorbed and detoxified by the liver. So it's something many scientists are working on, and I think it's a very exciting field. So increasing our knowledge on the human body and how we can respond to drug treatment. This also helps to understand this before we move into animal studies or clinical trials that require, of course, a lot more work. But 3D printing is not the only way to achieve the production of human organs in the laboratory. And I'm going to tell you more about the technique we use in the laboratory work now, which is called decellularisation. Decellularisation is a technique that has been developed quite a few years ago now to remove all the blood and the cells that an organ is made of. This process allows to have left the natural skeleton of the organ without the cells. That is basically a mesh of protein, very organized, that we call extracellular matrix. So it's really like a scaffolding that I've shown you before for developing like big skyscraper. So you see here a picture of the organ before the decellularization and after decellularization. And you see they basically become transparent, translucent. Sometimes you can see through them. And this is because we removed all cells, we removed all the blood, but we have left the 3D structure of the organ itself. And we have left, for example, sometimes even the vasculature. So this is the idea. So we have a liver, for example, the organ of my interest, we decellularize, removing all the cell, and then we can put back healthy cell. We repopulate the liver with a healthy cell. We have a new and healthy liver that we can use in the laboratory. That's what I usually do, to study how liver disease happen and to test the different drug treatments. But one day, there are many groups that study how to apply these for transplantation. And here I show you some data we produced in the laboratory of a liver before and after decellularization. And basically, we measured the DNA content before and after decellularization. Because we remove all the cells, and you know very well that the DNA is inside the cells, we show that we also were able to remove completely the DNA after decellularization. So no more cell, no more DNA. This is an image of a liver before decellularization, and you can see how dark it is. The pinkish is basically the presence of cells. And this is the scaffold, so what we call it after decellularization. You can see there is no pink basically left. There is just this structure here, which is the extracellular matrix, so the structure of the organ. And we put, after we decellularize, some blue ink in the vasculature of the liver, and we show that we maintain a beautiful vasculature tree. This is very important because we can use this vasculature to deliver nutrients and blood exactly as the organ has in the body. So it's very promising. So we got the cells, we got scaffold, either 3D printed or cellularized. How can we deliver good molecules for the organ to be functional? So an organ in the human body, you know very well, it receives nutrients and oxygen and molecules throughout the blood. And, oxygen with lungs gets oxygenated, and it goes in every single cell of our organ. But we need to provide the same oxygenation and the same nutrients to a tissue-engineered organ as well. Otherwise, the organ would just die. So nutrients and molecules, they're usually supplied in the laboratory by adding the culture medium. Culture medium is the pinkish, reddish solution, very sugary, that the cells of the organ are grown in. But when we talk about whole organ, if you think about a big piece of liver or a kidney or a heart, we cannot just simply dip it in culture medium. It's too big. It's not enough for the solution to go through and reach all cells. So we need to find a way to deliver the nutrient to this big construct. And it's not very easy. In fact, there is a lot of people that are thinking how to do this. And generally, scientists and engineers work together side by side to develop what we call bioreactors. Bioreactors allow to culture artificial organ for a very long period of time by providing oxygen and nutrients. And this is just an example, a few examples of how bioreactor look like. And you might think, oh my God, but they're all different from each other. And that's indeed one of the very fascinating things about this field, because we can produce different type of devices. We can adapt them to the actual structure of the organ itself. So for example, these 3 bioreactors here are used for the culture of whole organ, for example, intestine or the esophagus, which is the tube, the food pipe that connects mouth to stomach. Otherwise, we have another different design as a bioreactor for different non-hollowed organ. And this is a bioreactor we actually produced in our laboratory. This is the design of the chamber of the bioreactor. And here we put the liver in. So you can see here inside the incubator, this is our bioreactor. This is where we put the liver, and it's connected to a pump that perfuse nutrient and oxygen through the scaffold, through the liver, like basically the blood does. And we can have this bigger or smaller depending on the necessities and the experiments. So it's extremely fascinating. So now we got cells, we got scaffold, and we got a way to deliver molecules, so by a bioreactor. So I hope I convinced you that we have a possibility to develop organ in the laboratory. And this is very important because despite huge improvements that we have in medical care, there are some diseases that affect vital organs, such as, for example, the liver, which are not curable yet. And the only resolutive treatment is transplantation. However, we cannot transplant all the patients that are in need because of a shortage of donors. We are working very hard to try to make organ in the laboratory from scratch to solve this issue. 3D printing and the cellularized organ are both a very valid alternative, together with the use of stem cell and bioreactors that we believe will make the culture of organ for transplantation in laboratory possible. Tissue engineering is therefore a field that is moving extremely fast to try to tackle this issue and produce organs. And another fascinating thing about tissue engineering is that it's very interdisciplinary. We have biologists, engineers, doctors, and surgeons that are all working together to try to bring organ from the lab to the hospital. So you might be very interested in the topic, but not really a biology kind of person, but maybe a more an engineer person, very interested into the bioreactor thing. And you might be able to approach this field by a different angle. Maybe you want to become a surgeon and you could just try to study how to transplant this organ into patients. So it's very exciting and it's beautiful always to work with people that study something slightly different than you, and we all help each other. We all have the same questions and the same aims. So it's something very exciting. So I would like to thank STEM Learning and the Catalyst for inviting me to talk about this topic I'm very passionate about. I like to thank my lab members and colleagues, all the institute, and as a resource that I would like to suggest you to have a look at is Eurostem Cells. It's a website that provides a lot of information about stem cell. And actually, I found these very cute comics about stem cell, because stem cell can give rise to any cells. Stem cell parental advice would be you can be anything that you want to be when you grow up. And this is something that is always good to keep in mind. You can do anything in the future if you would like. And thank you very much for listening.
Thank you, Sara. We have some questions for you now from our audience. The first one asks, what subjects do you suggest studying before university in order to pursue this kind of research?
That's a very good question. Thank you very much for asking. I recommend study biology for sure. That will give you a great advantage at uni. If you know already, if you have an A-level biology study. But of course, any scientific subject would really help. So mathematics, physics, chemistry would definitely help you at uni for them going through the subject. Uni choices, I'll say any biomedicine or molecular biology courses are all good. We get students from mostly biomedicine, but biology as well, or biotechnology, so any of these.
Thank you. Our next question asks, can this revitalization of the liver be done in the human body?
Actually, that's a very, very good point. There are some machines at the moment that have been developing, but also studied in clinical trial to try to keep organ alive after a donor has gifted the organ for transplantation. Because most of the time the organs are not available for transplantation because, as you probably know, transplantation has to happen very quickly after the death of a donor. So what some machines are trying to do is to keep the organ alive. And some people are also studying like to keep these inside the body or outside the body. So definitely there is research there. In terms of what I've shown you today, so 3D printing of the organ or decellularisation, that has to be done outside the body, but there are other techniques to do that inside.
Thank you. Our next question is, what is your favorite part of your research?
Oh, there are many different things I love about research. One of the things I like the most is that every day is different from each other. So we have experiments most of the days, but those can be very different one day to another. And any day is really exciting. I don't know what's going to happen by the end of the day. Like could something extremely good come up from these experiments? Sometimes it's also not very good, but we have plans B and C to go through a bit more tough days, and is the fact that it's very diverse job really excites me and gives me the possibility to develop so many different skills, and I think it's wonderful.
How long do you think it will take for entirely lab-grown livers to be used in patient treatment?
This is a very difficult question to answer. Considering the huge progresses that have been made in the past years, I would say hopefully less than 10 years. However, we have to say that there are already some organs, for example, from the decellularized tissue being transplanted to patients. So from few organs, this time frame that I have already given might be even way shorter, might be matter of only a few years. Certain other organs, such as the liver or the heart, might take slightly longer, but hopefully for some less.
And for our final question from the audience, how do computational biologists fit in this workflow of research?
Oh, very nice question. Well, computational biologists are now really, really important in any side, any topic in biology. We do some computational analysis of our scaffold. So when we put cells in the cellularized tissue, we put different type of cells. And what we are trying to understand is how the cells interact with each other and compare these with the organ of original organ with a liver from humans. So we really try to combine normal classic biology study with computational analysis. This is what many groups are doing. So really they fit in perfectly because we have to have a reference and a constant comparison with a healthy liver. So we apply computational biology for that.
Absolutely amazing. Thank you very much.
Thank
well, that
you
was
very
the end of
much.
the questions. So very quickly, because I appreciate you have to disappear off to something
Sorry.
else. So thank you, Sara, for such an incredible exploration of your scientific research into tissue engineering, and in particular your work on human livers. It is very clear how important it is for scientists like yourself to push the boundaries of knowledge and seek to find solutions, particularly for transplant patients. Thank you for sharing your research with us. Sara has written an article on today's topic, which is available to read in Catalyst magazine. Please explore it at the link provided in the chat. And thank you to our audience for joining us today. The next event will be held in the autumn term, and we will publish details in September. We now hand over to Mary, who will showcase how today's topic can be used to support curricular learning.
Good afternoon, everybody, and thank you very much, Sara, for a really interesting talk. I think that's really inspirational. My name is Mary Howell, and I am the biology lead, professional development lead at STEM Learning, and I've got some resources that I would like to share with you this afternoon. And at the end of this, I will share a link to a resource collection that I've set up on STEM Learning's website, so that you can just use that one link and find all the things that you are looking for. So in fact, I will put that. So I'll put that in the chat later. There is quite a bit of overlap between the set of resources for today and the resources that Chris Carr shared after the Biomedical Sciences in Action Catalyst magazine live session. So what I've done is I've put a cross-link to those resources at the end of my resources list, and also a link to the video from that. And then I've tried to find some resources that complement today's talk without repeating what Chris said last time. So I'm going to share my screen, and one of the things that Sara talked about, was about the role of the liver and how important the liver is. And I thought a really useful curriculum link there is to look at diabetes and blood sugar levels, because although we don't do a topic perhaps on the liver, we do look at several things about drugs and homeostasis and diabetes and blood sugar levels in the curriculum, which strongly relate to that. And a set of resources that I really like are these ones from ABPI schools, which you can just search around. And if you look at the website, you'll see that there are resources for all age groups. A lot of them are study resources, and they have made some changes to this website recently. But if you go to this is a 14-16 resource, and if you go to this menu on the right hand side, you will see, if you click on that you get a list of page numbers of the study resource that you can work through. In addition, you get, if you click on the Venn diagram symbol, you get a list of related topics, and you can also click the summary, the question mark, and that will take you to a guide to how long it should take you to work through the resource, and also some information about the glossary of terms, which for a lot of students is a really helpful thing to look at. My top tip, if you're finding this without following the resources guide that we're going to put on STEM Learning's website, is to remember to put ABPI and then schools, because the ABPI do also produce academic papers and so on. So it stands for Association of British Pharmaceutical Industries. Another example of their resources, this is in the the citizenship and PSHE section, and there's a section here about cloning. And just to give you an idea of the breadth of what there is in biology, in the 14 to 16 and 16 plus section, there's a section about stem cells. What do stem cells do? What are stem cells? And if you look here, there are lots of links here that you can click, and also you can see that it describes the role of stem cells, it describes what differentiation of cells is, it describes what embryonic stem cells are, and it looks at adult stem cells. So really nice. And then before I leave ABPI completely, another thing that I really like is there are some really nice posters. So if you go to the link that I've put in here, then you can choose from several posters and they will send you physical copies of these. So in particular, relevant to here, to today's session, there is this very nice one called Stem Cells Horizons in Medicine. You can see that there's also some on genomics, polymerase chain reaction and genetic engineering, cloning, which again links really nicely to what we've been talking about today, and biotechnology. So ABPI schools are a really worthwhile site to have a look at and have an explore if you haven't seen that recently, or if you've used it in the past and then have forgotten about it. Another really useful resource, something that perhaps not everybody's come across before, is Wellcome. The Wellcome Trust have a Wellcome image collection. I have to admit that the way I find these resources is to search on a search engine rather than in the website itself. But if you type in Wellcome image collection, for example, IVF, and you can see you've got these really good quality images. You've got information about the copyright. Many of them are Creative Commons or have got agreements in place that allow you to use them, for example, in PowerPoints in schools. So you can find the images you like. But I think it's really helpful when you're talking about human stem cells to be able to talk about that idea of that ball of cells that develops from the fertilized egg, and to be able to talk about the idea that every cell has got a full set of the genetic information, and then talk about cell differentiation and work from there. So Wellcome collection, Wellcome image collection, lots of really nice resources there. Another site that I think is really useful and good to use, perhaps more for background and perhaps for your own, for teacher information as much as for students, but the Nobel Prize website, nobelprize.org. It has education resources for example, the 2021 Nobel Prizes and lesson plans. But in addition to that, for each year, you've got a list, you've got who the Nobel laureates are. So in 2012, they were Sir John Gurdon and Shinya Yamanaka, and their Nobel Prize for in physiology or medicine was for their work on stem cells, and the idea that you can take a mature cell and you can take a mature cell and you can reprogram it to start becoming pluripotent. If you scroll all the way down in the page, and you will find a link, and the link takes you to a really nice summary infographic, which, you know, for things like reading in class, literacy through science, making sense of diagrams. So there's some really nice information there in that infographic. Going a little bit further back in the history of the Nobel Prize, in 2007, there was a Nobel Prize for discoveries of principles about genetic modification in mice and the use of embryonic stem cells. And again, if you scroll down on that page, you will find a really nice infographic that has got some really useful information and a resource that you can download and share with your students to get them to think about that, but also to just think about where do these ideas come from. And something I particularly like about the Nobel site, if I go back to the 2007 page, is that how often it's a collaborative enterprise, that moving away from the idea of scientists working in isolation and the idea that they build on other people's ideas and other people's research, and that also scientists often work in teams. So an important aspect of science and understanding about careers and how science works. The next resource I'm going to talk about is available in two places. I couldn't get this link to STEM Learning's website to load into the collection that I was making, but there is a debate kit about stem cells, which it actually comes from I'm a Scientist, Get Me Out of Here. And if you're not familiar with that organisation, it's well worth a look, because each year they run an online debate similar to a balloon debate, where practicing scientists linked to a particular field of research are online, and students can ask them questions and they can respond. So you can sign up your students to take part in those debates online, and then the students vote, and every so often a scientist is eliminated, and eventually there's a winning scientist that has won the student vote. So those are worth looking at. But in addition, I'll just click on the next link to, if you go to the site, I'm a Scientist, Get Me Out of Here also has a series of debate kits. And one of the debate kits that's available is the Stem Cells debate kit. And if I click on that, she said, then that one is about, should the UK government invest in stem cell research? But you can see that there are things about food security, drugs in sport, and so on. So always a very nice thing to do. I think often teachers say that they perhaps don't have time to do an extended debate because of a crowded curriculum. But I think for motivation and engagement, it's really helpful. And I think it's a good way to help you to reach your remit for careers education, because of course, under the Gatsby guidelines and career benchmarks, that's a cross-curricular responsibility. So we often talk about putting examples into curricula and thinking about the roles of scientists or the roles of science in individual lives, but also thinking about those work-based learning skills. So, for example, thinking about communication skills, presenting an argument or a case, summarizing information, communicating, those sorts of things. So I think it's really helpful for those wider aspects of career education. Another useful site possibly not quite so obvious in terms of educational resources, but the NHS, the National Health Service, have some resources on organ donation and blood donation. These are pitched at Key Stage 3 and Key Stage 4. They're mostly tagged as being suitable for PSHE, but there is one on the science of blood donation. And these are presentations for assemblies or lesson plans for class use. And if we keep scrolling down, there are some things about donation and choices for PSHE lessons, and there are also links to charity partners of the NHS and some other resources, including some links to some things related to stem cells. So something worth a look if you're looking for something for the wider curriculum. And so that's the link to the resources from the charity partners. And I'll just scroll down. So you can see if you've got primary colleagues, or if you're teaching in a through school that goes from primary through all the way through to secondary, then you can see there's some things like organ donor support and information there. And then for 11 to 18-year-olds, there's We Are Donors, which is an organisation that recruits university students to work across UK's schools to run talks and so on. So people to deliver training. And so if you want something like that and some information, or you want to give that to perhaps a head of year or a tutor team lead, those might also be useful. But it's got some very nice cross-curricular links, okay? And then the Jemima Lazel Trust runs a creative writing competition to encourage young people from 5 to 18-year-olds to discuss organ donation. So there's I think the link there is a little bit out of date. It says 2019, but I'm sure that that's still running. Another thing that is useful to look at, another place to look, quite often I see people looking on Twitter for literacy resources in science or for wider reading to inspire post-16 students, for example. So the conversation is an arena where scientists and other people can put forward articles, doing things like summarizing their research and so on. You do, I think, need to read them and assess them for suitability for use in class, but they're written in easy to understand language. They're fairly short articles, and there's a range of those. I've just picked out this one, which is about genetic engineering transformed stem cells into working mini livers that extended the life of mice with liver disease. So something to have a look at, because you might find some resources. And that, I think, if you're interested in Catalyst Magazine and Catalyst Magazine Live, the chances are that you'll find those resources in the conversation, which is a nice, easy to search website to come up with articles on a range of topics. But there's some really interesting ones about tissue engineering and stem cell research, that go into the science behind it, as well as debating the ethics. BBC Science Focus magazine has got several articles, and again, you can find a way on here to download it as a PDF. This particular one is about mini organs grown for children with intestinal failure. So thinking about, what other examples are there? Is this actually something that's happening in reality, or is it still something that's in development? And also on Science Focus, an article here about scientists have grown a mini beating heart, or they call it an organoid, in the lab. And this is very recent, from May 2021. So well worth a look at those science-focused magazines. In terms of practical work, I know that Chris Carr mentioned this last time, but I'm going to mention it again. Cauliflower cloning, so Science and Plants for Schools, is a great website with lots of really good, manageable practicals that you can do in class. And this cauliflower cloning resource, you take a cauliflower head, the creamy white bit of the cauliflower, and you take a tiny floret and you soak it in Milton solution to sterilize it so it doesn't go moldy, and then you put it in a specialist medium and you can grow the organs of the plant, the leaves, the roots, the stem, from that little clump of cells. So although it might not be possible to do animal organ development and tissue culture and tissue engineering in class, you can definitely do plant tissue culture and engineering, and you can create these. And I think that's a great way to really, if I say wow factor, it implies it's kind of instant. And obviously, like a lot of biology, you have to wait for these things to grow and you have to be patient. But it's well worth the effort, and it just shows, and there's a video on here that takes you through the procedure, which is also well worth watching. I've included, as I said at the beginning, a link in the collection, the resource collection I've made, to the previous Catalyst magazine, Live, the Biomedical Science in Action. And in my link, I've said that if you want the bit where Chris is talking about the resources that he recommended, because there was a lot of overlap, the things that I had thought immediately when I was asked to do this would complement what Sara did. Chris had already mentioned, so I've cross-linked to that previous Biomedical Science in Action, Catalyst Live, and I've also, she said, cross-linked to the actual resources list that's on the STEM Learning website as well. So thank you very much for joining us, and I hope you found that useful, and I hope you enjoy exploring those resources for yourself and for your students. Thank you.